Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Kerala SET Biotechnology question paper conducted in the year January 2025. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of SET Biotechnology being discussed, you can go to my channel Omni Bioentrance. In the playlist you can see for the folder Kerala SET Biotechnology, and in there you will find. Coming to the next question, the 9th term of an arithmetic progression is 76 and the 19th term is 46. Then the term is, so an is equal to a1 plus n minus 1 d. So d is the common difference. A1 is the first term, an is the nth term. So here you can make two equations. So the 9th term is 76 and 19th term is 46. So substitute in the formula. 76 is equal to a1 plus 8d, that is 9 minus 1, so that is 8, that is 8d, and 46 is a1 plus 18d. So 46 is 19th term, so 19 minus 1, it is 18d, and upon subtraction you will get d as minus 3. Substitute the value of d in any of these equations and you will get the first term, that is 100. Now you got the first term, you can find the 30th term, substitute the values, and the answer is 13. Answer is option A. Then next one, the data set has a mean of 10 and a standard deviation of 2. If every item in the data is increased by 10, then the coefficient of variation of this set is. Now one thing I will tell you is if every item in the data is increased by 10, your standard deviation is not going to be affected because it is a constant. Now the mean will be increased by 10. So in this case the original mean is 10 and the original standard deviation is 2. The new mean, that is mu dash, will be 10 plus 10, that is 20, and standard deviation it will remain the same. So coefficient of variation is sigma by mu, where sigma is standard deviation by mu, that is your mean. So substitute, you will get 10% as the answer. So answer is A, 10%. Then next one, the oncogene associated with chronic myeloid leukemia. So chronic myeloid leukemia, it is as a result of chromosomal translocation between the 9th and the 22nd. And this result in a product that is BCR ABL fusion gene. Okay. And this BCR-ABL fusion protein, that is the product of this gene, that is the protein. It has tyrosine kinase activity and it can promote leukemia. So the oncogene associated with chronic myeloid leukemia is BCR-ABL, that is the oncogene. Then the primary function of G protein in cell signaling is, so DNA binding. G protein doesn't play a role in DNA binding, it plays a role in signal transduction. That is G protein transmit signal from the G protein coupled receptors or the GPCRs to downstream members. So it is signal transduction. Then which of the following about P10 tumor suppressor gene is incorrect? P10 regulates PI3K AKT signaling. Yes, it is a negative regulator of this particular pathway, that is PI3K AKT. Then P10 activates NF-K beta. Now that is not an function of the P10 tumor suppressor gene. So they have asked the incorrect statement. So it activates NF-kappa B is not correct. Then P10 promote apoptosis, yes, and it is a tumor suppressor. So it will help in preventing the tumor growth, so it can promote apoptosis, so that's correct. P10 role in inhibiting cell migration, yes, because cancer cells, they have a tendency to migrate or spread. So P10 again a tumor suppressor, so it inhibits cell migration, that is also correct. Then the purpose of F0, F1, F2 in Punnett square. So F0, what is this F0? It refers to the parental, that is F0. F1, progeny upon crossing these parents, that is first filial generation. F2 is again crossing the F1, you will get the second filial generation. So all these things you represent in the Punnett square, and what do you see? You get the various genotypic ratios. How many dihybrid cross of Mendel, if you see how many yellow round or wrinkled seeds, etc., etc., you can find. That is how many tall are there, how many dwarfs are there, all these you can find out. So it predict the genotypic ratios. So answer is option C. Then if a leucine oxotroph means you have to supplement the medium with leucine. Here it is leucine oxotroph, so you have to supplement the medium with leucine for that particular bacteria to grow. Is grown on medium supplemented with leucine. Okay. The strain will grow. It will grow normally. Answer is option A. You are providing an oxotroph with leucine, so naturally it will grow. Then next one. Dash is the primary function of hammerhead ribozyme. So ribozyme, it is for cleavage of RNA phosphodiester bond. So answer is option C. Then what is the role of aurora B kinase enzyme during mitosis? So I already discussed that, I think. Aurora B kinase, it is for correcting improper kinetochore microtubule attachments during mitosis. Okay. So the answer here is correction of kinetochore attachments. Answer is option B. Then Franklin's X-ray diffraction images, which was crucial for Watson-Crick's DNA model, it was photo 51. Then next one, the primary mechanism by which DNA polymerase ensures fidelity during replication. Fidelity is accuracy with which the DNA is copied. That is by minimizing errors. So how does DNA polymerase does this? It is through proofreading and editing activity of the DNA polymerase. So proofreading means it will check the newly added nucleotides and correct them if there is any mistake. Then editing also, if there is a mismatch detected, then the exonuclease activity of the DNA polymerase removes the incorrect nucleotide and bring in the correct one. So that is proofreading and editing. Complementary base pairing, it is required for accuracy, but it is not the mechanism for ensuring fidelity. And beta clamp, it is not an enzyme involved in fidelity, and helicase activity is for unwinding the DNA double helix, not for fidelity. Then next one, the ion channel involved in repolarization phase of action potential. So repolarization, it is the condition where the cell membrane potential returns to its resting state after the depolarization step. So it is driven by outflow of the potassium ions from the cell. So for that you need to open your voltage gated potassium channels. So the ion channel involved in repolarization is voltage gated potassium channels. Then next one. Plant adapted to dry environments are characterized by CAM photosynthesis, that is crassulacean acid metabolism. There is no crassulacean photosynthesis, it is CAM photosynthesis. Then identify the true statement about Km. Now Km, it is a measure of the affinity of an enzyme for its substrate. So low Km means high substrate affinity. High Km means low substrate affinity. So that makes statement 1 as wrong. Statement 1, what they are saying, high Km indicates high substrate affinity. No, it is low substrate affinity. Km changes with substrate concentration. So it doesn't change with substrate concentration. It is a constant and it just reflects the affinity of an enzyme for its substrate. And Km is of course the substrate concentration at which the reaction rate reaches half the maximum velocity or the Vmax. And it does not change with the varying substrate concentrations. Okay, so B is also not correct. Km changes with the enzyme concentration. No, it is a constant and it doesn't have anything to do with enzyme concentration. Km is independent of enzyme concentration. Yes. So the correct answer, they have asked for the true statement. So it is Km is independent of enzyme concentration is the right answer. The type of inhibition which occurs when the inhibitor binds to the enzyme substrate complex. Okay. Now if you see competitive inhibitor, it competes with the substrate binding site of the enzyme, or I can say it competes with the substrate for the active site of the enzyme. So that is what competitive inhibition do. It doesn't bind to enzyme substrate complex. Now non-competitive inhibitor, it binds to allosteric site on the enzyme, not to enzyme substrate complex. Uncompetitive inhibitor, yes, it binds to enzyme substrate complex and not to free enzyme. Okay. So C is the answer. Mixed inhibition, it can bind both to the enzyme as well as the enzyme substrate complex. So the answer here is option C. They have asked for the inhibitor which binds to the enzyme substrate complex. Okay. The type of biosensor which measures changes in the refractive index of a solution, it is surface plasmon resonance biosensor. Answer is option D. Then the primary location of LDH1 isoenzyme, it is found predominantly in the heart muscle. Answer is option B. Then which complex in the electron transport chain is responsible for pumping the most protons across the inner mitochondrial membrane? Now if you see the complex 2, it does not pump protons. It transfers electrons from succinate to ubiquinone. Other than that, it doesn't pump any protons. So B is not correct. Now cytochrome 3, that is BC1 complex, it pumps proton but it is fewer than the complex 1. And cytochrome 4 oxidase, it also pumps proton but its main function is to transfer electrons to oxygen. So the one which pumps the maximum, which one, what they have asked? Most protons. Okay, most proton is complex 1 NADH dehydrogenase. Then next one. The primary advantage of using creatine phosphate as an energy source during high intensity exercise. So creatine phosphate, it has a role in rapidly regenerating ATP, which is the primary energy currency of a cell. And it will because it will delay the onset of muscle fatigue. Okay. So they have asked primary advantage. Remember the word primary advantage. So it is rapid release of energy. Okay, there is increased endurance and enhanced recovery also, but they have asked primary advantage, it is rapid energy release. Then the half-life of a radioisotope, you are learning it from your high school. It is time taken for half the decay. So answer is option D. The exact function of matrix in Maldi-Toff is to. Okay. So it is absorb excess laser energy and facilitate sample ionization. And upon this ionization, they can be analyzed by the mass spectrometer. So that is the main function of matrix in Maldi-Toff. So answer is D. The process of adjusting the FACS, that is fluorescence activated cell sorting instrument, to optimize separation of cell is termed as, it is called as gating. So here this FACS, it is a flow cytometry that sorts a mixture of cells based upon specific light scattering and fluorescent characteristics of the cells. Now what is this compensation? So that is what FACS do. So to optimize separation of cells, it is termed as gating. So what is this compensation? So now this different fluorophores, they emit light at different wavelength, and these emission spectra, they can overlap in certain cases. So when this happens, the light emitted by one fluorophore, it can be detected by multiple detectors if your instrument is having multiple detectors. So, and this detection by multiple detectors is called as spillover. And this spillover, it can lead to inaccurate data, and the solution for this correction of this spillover is by compensation, and this compensation, it uses mathematical algorithm to correct this spillover. And optimization, it refers to adjusting various experimental parameters such as sample preparation optimization. That is, you have to create a single cell suspension so that there will be proper detection and sorting of the cells. Then staining optimization, that is, you have to choose the right fluorophores and at what concentration they have to be used. Then there is sorting optimization, that is by adjusting the detector voltage so that you can have better resolution, and then there is a data analysis optimization. Okay, then what is calibration? Calibration is adjust the instrument to produce accurate and reliable measurements. That is calibration. Then next one. In gas chromatography, what would be the effect of increasing the temperature of the column? So these higher temperatures, they increase the volatility of your analytes. So that will reduce the interaction of the analyte with the stationary phase. Okay, that is, your retention time is reduced. And this will cause, what will happen when the retention time is reduced? It will elute faster. So effect of increasing temperature of the column in gas chromatography, there is decreased retention time. Then a pharmaceutical company need to formulate a liposomal drug delivery system. Which centrifugation method will you adopt to determine the size of the liposomes? Of course, it is going to be dynamic light scattering, and some of you will have a doubt with regarding to ultracentrifugation. Yes, but it is not just simple ultracentrifugation that you have to use. You have to go for the analytical ultracentrifugation, because the normal ultracentrifugation, it is used for separation and purification of particles, whereas analytical ultracentrifugation, it uses optical detection method, and it gives information about the size, shape and mass of the molecules. So answer is option C. Then next one, name the radioisotope commonly used for cancer treatment due to its high energy emission. It is cobalt 60. Then measles and Stahl experiment in 1958 utilized. So measles and Stahl experiment, you know that it supported the DNA hypothesis that DNA replication is semi-conservative. So they have used N14 and N15 labeling and cesium chloride density gradient centrifugation method. So the answer here is option B, N15 labeling and CaCl2 density gradient centrifugation. Then which of the following is a characteristic of inducible operon? Constant expression. No, there is no constant expression in case of inducible operon. Only when the signal is there, the operon will be induced and the enzyme will be produced. So expression is triggered by specific inducer molecule. That is correct. Answer is B. Then post-translational modification that removes N-terminal methionine from nascent protein. So it is by the process called as aminopeptidation, and it is carried out by the enzyme methionine aminopeptidase. Okay. Now deacetylation, you know, it is removal of acetyl groups, not methionine. Then demethylation. Demethylation, it is for removal of methyl groups, not methionine. Okay. Then next one. The viral protein responsible for mediating the attachment of influenza A virus to the host cell, that is hemagglutinin. Matrix protein, it is a structural protein that is found beneath the viral envelope, and it is involved in viral assembly and all. Then nucleoprotein, it encapsulates the viral RNA involved in replication, transcription, like that. Then neuraminidase, it is also another surface protein, and it is responsible for cleaving the sialic acid receptors to facilitate viral entry. Now hemagglutinin, what it does is it mediates the attachment of influenza virus to the host cell by binding to sialic acid receptors. And neuraminidase, it cleaves the sialic acid receptors and it will help in viral entry. So that is, here they have asked mediating attachment of influenza A virus to host cell. So it is hemagglutinin. Then which of the following is not considered a functional food? So functional foods, it can be your fruits, vegetables, fortified foods, that is naturally fortified with beneficial compounds. Okay, so they are functional foods. Probiotics are functional food, dietary fiber is functional food, omega-3 PUFA is also functional food, but pills is not functional food. It is, pills are isolated nutrients or compounds in concentrated form. So it can be your vitamins, your minerals or herbal extracts. They are not functional foods. So the answer here is option C. Then the expression enzyme system used to produce recombinant human atyplase. So atyplase, it is a recombinant protein and it is used in the treatment of strokes and what is that, break down blood clots. For all these things you use ATPase. So it works by conversion of plasminogen to plasmin. So the expression system that was used to produce this recombinant human ATPase is CHO, that is Chinese hamster ovary cells. E. coli cells are usually not used for proteins which require post-translational modifications. Okay. So yeast also they can be used for protein expression of proteins which require post-translational modification, but the human ATPase it requires complex modification, so it will not be used. HEK293 it is human embryonic kidney cell, but for the recombinant human ATPase you use CHO, that is Chinese hamster ovary cells. Then the microbial metabolite which plays a key role in regulating host immune response, it is your short chain fatty acids. So they are involved in reducing inflammation. Then they influence the activity and differentiation of T cells, dendritic cells. Okay, and they support the health of gut epithelial lining. Whereas peptidoglycan bacterial cell wall component and it triggers immune system. Then lipopolysaccharide found on the outer membrane of gram-negative bacteria, it is also stimulates the immune response. Indole 3 propionic acid, it also influences immune response. So here key role in regulating host immune response is short chain fatty acids. The mechanism by which recombinant TPA, that is tissue plasminogen activator, that is your ATPase, enhances thrombolysis in acute ischemic stroke is it is not inhibiting plasminogen, it is activating plasminogen. So first is not correct. Enhancing fibrinolysis through plasmin generation, yes. So TPA binds to blood clots, activates plasminogen, converting it into plasmin, and this plasmin it will break down the fibrin, thereby dissolving the blood clot. Then inhibiting platelet aggregation, that is not function of TPA. Stimulating endothelial nitric oxide production, it is also not a function of TPA. So answer is option B. The drug which inhibits platelet aggregation by inhibiting the enzyme cyclooxygenase and formation of thromboxane A2. So it is aspirin, and it inhibits the enzyme cyclooxygenase in platelets, and this inhibition it will block the conversion of arachidonic acid to prostaglandin, and prostaglandin it is a precursor of thromboxane A2. So when there is a block in the production of prostaglandin, so there is a reduction in thromboxane A2. So when there is reduction in thromboxane A2, there will be decrease in platelet aggregation and vasoconstriction. Okay. So answer here is option A, that is aspirin.